Phytoplankton are tiny photosynthesizing organisms found in the ocean's sunlit surface, where they underpin the marine food chain, provide the world with oxygen, and through their birth, growth, death and decay, they play an essential role in the global carbon cycle. Despite their small size, they account for approximately half of the global net primary production. Phytoplankton are particularly sensitive to changing environmental conditions. Therefore, climate change may have severe implications on the distribution, abundance, and seasonality of phytoplankton. The most effective means of monitoring phytoplankton is through ocean color satellite imagery, which provides a synoptic view of the ocean. Unlike in situ samplings, remote sensing ocean color imagery offers an unparalleled tool for examining phytoplankton dynamics by providing extensive global coverage at high spatial and temporal resolutions. In the Spectral Remote Sensing Laboratory, under the supervision of Maicero Costa, researchers and students use satellite imagery to uncover the intricacies of phytoplankton dynamics in the coastal oceans of British Columbia and Southeast Alaska, a region that encompasses essential habitats that support a diversity of economically and culturally important fish stocks such as Pacific salmon. In light of rapid climate change and its predicted impact on marine ecosystems, understanding the spatiotemporal patterns of phytoplankton blooms and their triggering factors over this region is of vital importance, both retrospectively and in a forecasting context. Phytoplankton phenology reveals the interannual and decadal variability in the timing of phytoplankton blooms, which is of particular concern because any changes at the base of the food web can have severe repercussions for the entire ecosystem. Cushing's match-mismatch hypothesis states that the timing of food availability may be as important or even more so than the abundance of food. The reason for that is when the timing of phytoplankton blooms fluctuates in response to changing environmental conditions, it can cause a decrease in the synchrony between the timing of food availability and the critical life stages of higher trophic levels, leading to an overall reduction in their survival rate. MSc student Sejal Pramlal is currently using satellite data to define the changes in phytoplankton phenology over the BC coast since 1998. Phytoplankton phenological indices such as timings of initiation, peak, and duration are used to characterize phytoplankton growing periods. Additionally, to examine the link between phytoplankton and zooplankton phenology in the Strait of Georgia, postdoctoral researcher Dr. Karan Suki is combining remote sensing data with a historical zooplankton dataset from the Institute of Ocean Sciences. By focusing on the biomass-dominant zooplankton groups that are important prey items for juvenile salmon and herring, this work will determine how the synchrony between phytoplankton and zooplankton varies in response to different environmental conditions, and how these changes may impact the food available for higher trophic levels in the region. Nowadays, there is an urgent need to establish representative monitoring regions that may optimize observation programs. Bioregionalization using remotely sensed data presents a valuable tool to address this problem. However, the currently existing large-scale bioregionalizations of marine environments are too coarse to provide a robust framework to interpret key ocean processes. To fill this gap and objectively define biogeochemical provinces across the coastal oceans of British Columbia and Southeast Alaska, postdoctoral researcher Christian Marchese uses an unsupervised machine learning approach to self-organize validated Sentinel-3 derived chlorophyll A data. The delineation of bioregions at regional scale represents an essential step to assessing changes in ecosystem structure and can be a relevant area-based management tool for fisheries ecology and ocean management. Beyond deriving chlorophyll A concentrations, satellite imagery is also used to retrieve phytoplankton functional types to help us understand the role of phytoplankton groups in ecosystem functioning. Phytoplankton functional types are groupings of phytoplankton species which have common ecological functionalities. Examples include phytocysts as DMS producers, diatoms as silicifiers, some cyanobacteria that are nitrogen fixers, and coccolithophorids as calcifiers. In this regard, PhD candidate Vishnu P. Susilan is combining ocean color and HPLC pigment data to derive phytoplankton functional types across the British Columbia coastal waters. To retrieve chlorophyll A and phytoplankton functional types, the reflected light from the ocean, measured by satellites, has to be properly corrected to eliminate the atmospheric signal. The SPECTRAL lab, together with Ocean Networks Canada, installed 2 sets of Satlantic Seabird SAS solar trackers on top of the commercial BC ferries to measure the in-situ ocean reflectance as the ferry crosses the Salish Sea. PhD student Ziwei Wang developed protocols to correct for superstructure perturbation and non-nadir viewing geometry, and quality assurance methods to make sure acquired data can be used to aid the validation of satellite-measured ocean reflectance. Postdoctoral researcher Dr. Naziha Jahir Hussain is further applying this dataset to understand the dynamics of the Fraser estuarine plume waters. A better understanding of the links between phytoplankton dynamics and environmental conditions is essential to predict the potential responses of phytoplankton to natural variability and climate change in the future. Therefore, this research program lays the groundwork for future studies examining how variations in bottom-up processes might impact the entire food web, which can inform future decision-making for sustainable fishing management and conservation efforts. As researchers who live, play, and learn on Indigenous land, we acknowledge and respect the many nations on whose traditional territory we work, and whose historical relationship with the land and sea continue to this day. We would also like to thank our collaborators, partners, and funders for making this research possible.